Now to the next talk of our conference. It's my pleasure to introduce a good friend of mine, Stefan Linquist, who's also here in Canada in the University of Guelph. And he will be telling us about, as you can see on the slide, epigenetic this, epigenetic that, and I'm very interested to hear about the rest of it. Stefan, take it from there.
Great. Thank you very much, Hekup. And, I wanted to just say at the beginning, I really appreciate your work and Jamie's work in organizing this conference. It's good to have a community of people who work on these kinds of projects. So thank you for that. I wanted to just also mention that there are three authors on this. This paper will be forthcoming in Synthèse. You can get a copy of it on my biophilosophy website here. And so Brady Fullerton is a PhD student at the University of Guelph, and he's got a really interesting data project that hopefully you'll be hearing about soon as well. And Akashdeep Grewal is a student who had a lot of expertise in the biomedical sciences, who we employed, and then he continued on with the project, and now he's in medical school. So like many philosophers of biology, I suspect, I've been long under the impression that the biological sciences are in the grip of some kind of epigenetic revolution. And what I took that to mean is something like this, that there's been a movement away from genes as the central unit of investigation and explanation. And in their place, there's been a bunch of other non-genic entities inside the cell that have similar causal roles, so influencing development and evolution. So why did I believe that? Well, I guess because I was under the influence of some very, established authors who've been influential in my discipline. So Susan Oyama, Evelyn Fox-Keller, Eva Jablonka, and Marion Lamb. About 10 years ago, I started collaborating with some molecular evolutionary biologists, and I ended up getting a different picture of what was going on with epigenetics. Some of them would use that term to describe the work they were doing, but they did not generally see it as revolutionary. And many of them saw it as very much continuous with sort of good old-fashioned molecular genetics. So here I was, influenced at one stage of my career by a small group of thinkers to think one thing about epigenetics, and then later on, influenced by another small group of thinkers. So one of the influential people in this field is David Haig, and he's written a lot about the meaning of epigenetics. And this is a nice very elegant study that he did on the frequency of use of the term as it appears in titles of papers. And he points out that it hung around for a long time, from the 1950s to the 1990s at a low frequency. And then in the late 90s, 2000s, the frequency of the term in titles of papers just exploded. And so one of the things that we're trying to do is drill into this a little bit. So here's an outline of the rest of the talk. I'll talk about the corpus that we put together in a moment. We analyzed it in two ways. We did a qualitative coding study and a topic modeling study of the exact same corpus. So that allowed us to compare the methods. And I'm going to argue that what we did is we converged on two conceptions of epigenetics, of what I'm calling a proximal conception and an evolutionary conception. All right, so we had a hunch that there might be some differences among disciplines in understanding of epigenetics. And as a way of operationally defining disciplines, we relied on subject categories supplied by the Web of Science. General biology is not really, what these are essentially are types of journals in which publications appear. So general biology is general journals. I'm not counting that as a discipline. It's more of like a control variable, if you will. But biomedicine, that's our category of a bunch of biomedical journals. And proximal biology, also a category that we constructed of some molecular biology and developmental biology journals. And evolution was a standalone category supplied by the Web of Science. And we wanted a temporal dimension to this. So what we did is we took the 50 most frequently cited papers from each of these 5-year time periods, okay? And so that's our corpus. And we looked, so we looked just at the abstracts, and I want to explain the reasons for that in a moment. Actually, before I get to that, so we looked just at the abstracts, and we did that for a variety of practical reasons. You know, there was a lot of reading to be involved here, and also sometimes within a paper, authors aren't entirely consistent in their use of a term, but you know, a lot of scientists only read the abstracts of papers, and we went with the assumption that that's going to be what they're most committed to, right? Whatever statements they make in the abstract of a paper is what they're willing to really put forward, as opposed to something that might appear at the end of a discussion, okay? Also, just for analysis, an abstract is way easier to work with. We had a lot of problems doing topic modeling with longer papers. All right, so even when you come to an abstract and you want to figure out, you want to answer questions like, well, do they think that epigenetics is committed to some kind of heritability? What sort of functional ascriptions are they making? You need to be a little bit more precise, because sometimes they're talking not about the epigenetic factors, but about the genes, right? How does an epigenetic factor affect the function of a gene or the heritability of a gene? So we needed to have a focus, and we focused on epigenetic marks as a category. And this is like an open-ended list of structures that interact with DNA. So then we came up with some heritability commitments, okay? So NA is basically, we couldn't identify an epigenetic mark being described in the abstract. Bare marks is a slightly, so then they would talk about a mark, but they wouldn't ascribe any kind of heritability commitment to it. A slightly stronger commitment would be mitotic inheritance transmitted from cell to cell, for example, during development. A stronger commitment than that would be limited meiotic. And so that's parent to offspring with a limit of 3 generations. And then open-ended is an even stronger form of transgenerational for indefinitely many, transmitted for indefinitely many generations. So we classified each abstract as having some kind of heritability commitment and also some kind of functional commitment. So here it's not quite as linear. Disease, transposon suppression, gene regulation, and adaptive plasticity. What do I mean by adaptive plasticity? Well, there's an idea that epigenetic marks help an organism over its developmental time adapt to changes in its environment. So they kind of make it plastic in a way that benefits the organism. I'm not too committed to this sort of ordinal ranking of these categories, but I will say that both of these bolder colored ones at the bottom are the most controversial claims within the biological sciences. And that is something I've defended elsewhere in print. So, okay, so here's what happens. You get a snapshot like this. And so here's general biology. And I'm going to go through this a little bit quickly. So over time, there was a sort of decline in the NAs. And what that was an indication to us of is that there was an increasing discussion of bare marks. And general biology is kind of a control variable. And you see a nice mixture of these different heritability commitments. Looking at proximal biology and biomedicine, there was a lot of similarity in the heritability commitments of these 2 disciplines, and they're both to basically very minimal commitments, mostly to bare marks. Some mitotic inheritance in the bolder orange, in the lighter orange, limited meiotic, very little discussion of open-ended inheritance, okay? And that's in contrast to evolutionary biology. So evolutionary biology, you really see this early on. They weren't talking about marks in the early mid-90s. They gradually started talking more about these specific cellular structures, but they often assigned to them higher degrees, more controversial ideas about heritability. So this is the top 50 most cited papers. And if you look at the top 25, the commitment to open-ended inheritance is even more significant. So in these papers, across these years, there were 30% of them would be committed to open-ended inheritance. Functional interpretations. So here's general biology. Again, you see this signal where they weren't always talking about marks, but they gradually did. Lots of talk about gene regulation, a little bit of talk about adaptive plasticity. Again, proximal biology and biomedicine are kind of talking the same language about epigenetic function. And there's kind of an interesting, we removed any entries that were identical in these two data sets, of course. There's an interesting sort of pattern here where disease kind of fell out of favor and then became more popular. A little bit of talk of open-ended adaptive plasticity, but not much. Again, evolution, a very different sort of discussion happening. An increasing discussion of adaptive plasticity. Some talk of regulation, but adaptive plasticity just seems to be becoming increasingly popular. So to summarize this part of it, so there's a growing emphasis on molecular structures, evidenced by this declining NA. These two disciplines, proximal biology and biomedicine, seem to be speaking the same language, but evolution's an outlier. I'm going to skip over some of the methodological lessons and maybe return to them later so that we can talk a bit about the topic modeling. We wanted to capture this temporal dimension. And so the way we tried to do that is but of course, each of these little data sets really weren't worth topic modeling on their own. So we lumped an earlier set of abstracts together and topic modeled them separately from a later set of abstracts. Okay, so for each discipline of the 4 disciplines, we have 2 topic models, earlier and later. And we assigned a coherence score to each of the topic models to determine how many topics to assign. We can talk about that later, up to a maximum of 25. Each of us who have different backgrounds independently assigned labels to them. And then we got together and compared our labels and really would only assign ones that we had a consensus agreement about. So look, I'm going to invite you to just squint with me for a bit, right? I'm not going to go into a lot of details here, but look, here's general biology, lots of stuff on methylation and gene regulation, some methodological topics in the earlier papers. Phenotypic plasticity is sort of out here. And you remember how it was like a small proportion across the years in the other study too? So it's kind of nice the way that the clustering here captured and even actually segregated those 2 discussions. Similarly, in the more recent papers, here's phenotypic plasticity in its own little satellite, and lots of discussion here about gene regulation. This is biomedicine. I'm not going to get into it. It's about cancer, okay? Lots and lots of discussion about cancer, a few more diseases in the more recent work. But I want to mention one thing in the proximal biology that jumped out that was just interesting. So here are the earlier papers on earlier abstracts on proximal biology. The main topic we see here, not a surprise, regulation and genome structure, then disease. There's a little bit of heritability here in parental effects and germline development. So that's kind of interesting. Lots of stuff on disease and regulation. Here's what surprised me. In the more recent papers, you've got regulation, some of these other factors, but the number 1 topic is this one called what we call biomedical promise. And let me give you a little insight into this. So there were words like this, these sort of promotional words like drug discovery, disease therapy, future health, and promise. Words like this just didn't show up anywhere else in the topic modeling. And it was really surprising to see them jump to the number 1 topic in these proximal biology abstracts. And this kind of goes along a bit with a criticism that's been made about epigenetics, that it's something of a buzzword that's being used these days to sort of mainly for promotional reasons, and that it doesn't have as much meaningful scientific content as it does sort of have a kind of a promotional role to play. So that was an interesting finding for us. Evolution, there's a few things here. Earlier papers talked a lot about Waddington, and I don't know if I have time to get into why that's significant, but he was one of the people who coined the phrase. And what you see in the earlier papers is a discussion of Waddington's ideas, and they disappear in the more recent papers. But what you see a lot of here is discussion of phenotypic plasticity, ways in which environmental factors purportedly affect the development of phenotypes in an adaptive way. It really is what the discussion is about in the evolutionary discipline of epigenetics these days. So patterns, well, it looks like there's a persistent emphasis in the topic modeling on regulation and disease in proximal and biology and biomedicine. There's little mention of heritability in these fields, though it did show up in proximal biology to some extent. So there's some discussion of heritability, but not much. So that's consistent with the findings in the other study as well. An increasing emphasis on adaptive plasticity, I'd say it shows up even stronger in the topic models, and an increasing emphasis on what I'm going to call biomedical salesmanship, if I can, in the proximal biology papers. Now, a brief comment or two on methodological lessons, and I'm almost finished. So I think that the topic modeling was a useful corroboration of the qualitative coding study. There were some striking things that showed up in the topic models that we found in the other study. So the extent to which certain discussions were segregated into different topics was kind of reassuring. But I've reflected a bit on, suppose we didn't find those things. Could you really falsify a hypothesis with a topic model? And I worry about my hunch is that not really so much. It wouldn't be a strong test because they really are amenable to different interpretations. So what I like to say is that they're better for generating hypotheses than they are for testing them. They can identify surprising patterns in the data. And I would say if you're interested in some scientific subject, it'd be a good idea to do both of these things. I also want to make sure I mention that when we were interpreting our topic models, it really made a difference that we had different background expertise, much more so than we did the qualitative coding, okay? The coding, so we did a replication study on the coding, and Cash has different background, but because those categories are so well defined, there was a lot of consistency in how we applied them. All right, that makes sense. But when it comes to interpreting the word list in the topic model, there were a lot of things that Akash would see because he's so familiar with the mechanisms, and he had a lot more to say than we did, who have, not as deep a background. So if I'm going to do topic modeling from now on, I think what I'll probably do is invite experts to interpret it with me, like, here's some word lists. What do you apply a label to it, please? So I think that'd be a good thing to do. So, all right, so general conclusions. So I think that there's a proximate sense of epigenetic, and it goes something like this, that there's a growing list of molecular structures that are thought to be involved in gene regulation and disease. But, there's minimal commitment to their heritability. Okay, there really just isn't much discussion of it, whether they're heritable or not. But there's an evolutionary sense that often talks about the same structures, but it views them as involved in adaptive phenotypic plasticity. So it's a stronger commitment to their function and takes them to have open-ended inheritance. So it's a stronger commitment to their heritability as well. Here's a conclusion I'm going to talk a little bit more about, and I'll be done. I think that philosophers and historians like me need to be wary of what I'm going to call the disciplinary founder effect. So this idea of the founder effect, that's something that is discussed in population genetics, right? Where you have a small population that founds a new, they're the new founding population in a new area. And since they're a subset of the home population, they're not representative. It's basically a non-representative founding population. And I think this happens in disciplines Let me give you one more graph, okay? So this actually is big data. This is from a 2021 paper that Brady and I published. And what we have here is the entire Web of Science mentions of epigenetics by the 4 disciplines we've been talking about as a frequency of mentions of DNA. Okay, so if a paper mentioned DNA, then some subset of those mentioned epigenetics. And really what you see here is, early adoption by molecular biology, followed by general biology and biomedicine. And here's evolutionary biology. They just weren't talking about epigenetics for about another 10 years. And even then, it's been a relatively minor topic, right? Even recently, it's much less pronounced. So what you get when you triangulate on all these things is that epigenetics is kind of a small topic. It's a small theme in evolutionary biology. It's very popular in these other areas. And when they do talk about it, they're talking about something really different from what the molecular biologists and the biomedical people are talking about, okay? And if we as philosophers get our sample from these people here and look over and go, oh, look, everybody's talking about epigenetics. What does it mean? Or what these people, we'd be really misguided if we took their definitions of it as representative of what was going on over here. So that's the lesson, right, that I wanted to make sure I was conveying. So again, Brady and Akash are co-authors on this paper. If you want to, I've put a draft of it here, and I've also got all, 8 of our topic models that you can interact with in an HTML format on my website there. And there are a few other people that were helpful in getting this paper to publication, including Ford Doolittle, David Haig, and Charles Pence. All right, thank you.
Thank you very much, Stefan. That was very interesting. I learned a lot. I cannot say I know much about the field specifically, but this looks very, very intriguing. Questions? Nicola.
Thank you for this other great presentation. Actually, I have partly a question, partly a remark, because about the biological promise topic in proximal in the proximal biology corpus, because I was struck by the similarity between the language associated with that topic and a kind of language that might be found in discussions about genomics and its possible application to personalized medicine. Therefore, I was wondering whether we could speak of a convergent evolution towards salesmanship in those two fields that might seem to be on opposite sides of the life sciences today.
That's really interesting. I think that one of my next projects is going to be talking about hype in genomics and trying to get, everybody hypes their work and says, I'm going to cure cancer, right? And what I've become very interested in is, when is the hype just too much? When is it being sort of deceptive to funders, for example? So maybe we can talk more about that. If there's a case of convergence, that's always interesting, right? That was your comment. Did you have a question?
No, it was that.
Okay, all right, thanks.
Thank you. Christoph?
Yeah, thanks, Stefan. Great work that you've been doing.
Thanks.
I really like the craft, the sales craftsmanship stuff and the hype thing. Definitely see that there is something there, even in the astrobiology community that I study very carefully. There is a lot of hype about typically finding, traces of life on Mars and other planets. So there's, this hype stuff is definitely very, very present also in this domain. But I also like your point about the founder effect that I had noticed in other domains, like in related to cancer, philosophy of cancer, and cancer approaches. Because it's true that sometimes in philosophy we are influenced by some very vocal persons, let's put it this way, who maybe have a strong thing, a strong message to convey. This message is not well received in their community, and so they go somewhere else, like in the philosophy of science area. And philosophers are very, interested and very happy to welcome scientists, because they not always and sometimes do not get this type of welcoming feeling. And we may get trapped into having a very, narrow view on some questions. And so I guess this is what you're pointing at with the with framing about epigenetics. But so I'm very sympathetic to this to these 2 points. I had a question about, when you mentioned the topic model. Typically, what we've done in the past was, in terms of for interpreting the topic models, definitely you can look at the top words, the top 10, the top 20. You can look at more than that, the top 50 words, just to make sure. And of course, the distribution of words will change depending on the number of topics that you have. So here, you can check do some sort of a robustness analysis by running different models and see if all of them are interpretable as easily as some others. But what we found extremely useful, in addition to having some expert knowledge, is to look at the papers, at the top papers. And very often, looking at the details of the top papers help a lot. So did you, because you didn't mention that.
But no, I didn't, but it happened during those collaborative discussions when we would come back together. We would, there were quite a few occasions where we would say, look, I don't know, let's look at the papers because we're in some disagreement here. So that ended up being in our practice, a kind of, when we would get stuck, we would do that. And I would say it happened about just 10-15% of the time. So it wasn't a systematic practice, unfortunately. I mean, it might be beneficial to do that, because you can actually identify representative papers, right? Like this is central to the topic. And so sometimes we would pull those out and go, oh, right, it's about the evolution of cranium something or other in this. And so, or a particular animal model would be sort of highly discussed. And so you could see that would come up once in a while. It would be interesting. I think there needs to be work done in sort of the systematic nature of these interpretations. And perhaps that is an element of that should be a stage. Look at X number of very representative papers. So we used it a bit, but not as much as we could have.
Yeah, we found it extremely useful, actually. And we do it now more systematically to at least recover the top 20 papers and scan them. I mean, depending on the size of the model, obviously, if you do a very fine-grained modeling, you may not do that for all topics. But it really depends on the research question. Now, the other point was here you did the topic model on only the few abstracts. So I understand the reason why you chose some random samples per time period, because you wanted to do some manual annotations. And you can definitely also do the topic, as you did, the topic model on the 2 subgroups here, but you could have done the topic model on a much bigger set of abstracts, because there is no limit there. And actually you could do that in addition to your small size topic model, just to see if there is some robustness there, if the the random sampling was appropriate. It could be a way of checking. That's a great suggestion. It wasn't random sampling, by the way. It was the topmost 50 cited papers. So correct. And the reason we did that is because and I didn't mention this, but I want to mention it now, that we're assuming that that citation is a form of approval. Unlike philosophers, scientists don't cite things they disagree with. Okay, not these days anyway. So that we took this as the more influential, a representation of a non-random representation of what's going on in the discipline. But I take your point. Look, you got to get a bigger topic model going. One thing we did is we did take all of our abstracts, put them in one topic model, and we wanted to see if sort of disciplines showed up.
we couldn't discern discipline. That was sort of one thing. Maybe disciplines will just fall out and we could get a corroboration on that. They didn't really fall out in any obvious way. Maybe they would with an even larger model though. And definitely there's work to be done now that we've taken a sort of baby step really to help us understand how the topic model relates to other types of analysis. I see this as a way forward to do more rigorous topic modeling.
And if I may, I have one last comment on the use the labels, the Web of Science categories.
Yeah.
I'm no longer certain, but is the because we played with different categories by different from different places. So the Web of Science category is only one label per journal, or
No,
are there several labels per journal?
It's many to many, as far as I can tell. So you've got labels that have many journals under them.
Yeah.
Okay, and some journals will appear in more than one label. Okay, so it's a little bit, so it's a little bit it's pretty rough, and I think there's a real issue here about if one should even take disciplines as a unit of analysis like this, and how you individuate them, and my answers to that right now are yes, you should, because they're real. I think they're real, and you try and get at them as many ways as possible and see if there's convergence. But this was just one way of doing it. They didn't rely on us. We wanted somebody else kind of making some categorizations. But
the thing is, in this particular case, you may have one journal that has several labels.
Yes.
And so, you're going to have potentially redundancies or overlaps between the groups that you set, because you did, biology, general biology, biomed, proximal.
Mm-hmm.
But there is, de facto, there is overlap here. So categories are not clean. They're not crisp, right? And so we encountered the same issue in another study in which you said, well, actually, we have to dump the labels because the labels are, vestige of the past, old database type of structure, idea of, being able to structure things. They may represent a little bit of something, but sometimes, just all the labels. Say you have one journal and it has 4 labels. Maybe one label is representative of 80%, and the other labels are only representative a little bit. So we dump all of them. And we did topic model to cluster the articles in sensible ways somehow, and then we disciplinarily interpreted these clusters. Okay.
Yeah, I think that it's another way to go, and it sounds really interesting. I'll have to look at your work on that. But what you might do is you might sort of approach the whole question with a different way of individuating disciplines, and then see how sensitive are these conclusions here to that disciplinary ontology. And my hope is that they're not totally too sensitive to it, but that's an empirical question, right?
Thanks all. Great work.
Thank you very much, Christoph and Stefan. Once again, another round of applause, Stefan.
Thank you.
Thank you very much.